Okay. All right. Good morning. I apologize for the delays in getting started. Had a bit of an emergency this morning. So let me go ahead and make a few announcements before we get started. So you took your third lecture exam and those grades have been posted. It is on my gradebook. I have to go in and update all of the mastering A&P part of it for your unit 3, but I can do that this weekend. Okay, so we've got today, and then we've got 2 more weeks of class where we will cover new content. And then after those 2 weeks, it is finals week. So during finals week, we will not cover anything new. It is reserved entirely for CTC students to complete their last set of exams. Some classes have final exams. We don't. Okay, we will just take our 4th unit lecture exam that week. So we have to follow CTC final exam schedules. So that's why, I mean, there's no new content, right? So we got to wrap up everything in the next 2 weeks, which we will be able to do. That's fine. Likewise, in lab, you all are also wrapping up the last unit for lab. And you should be taking that last lab exam as well right before finals week. So there's two big exams coming up right in the next couple of weeks. So a lot happening. All right. What else? I think we're good. And am I missing anything else? I think we're good. Yeah. Okay. All right. For this last unit, we're going to have to cover water balance and the role of sodium, potassium, calcium. How do you regulate those? How do you regulate hydrogen ions in relation to pH and acid-base balance? So that's what this chapter 26 is about. A lot of what is covered here, we've already discussed throughout the semester. So we're gonna review a lot of these concepts. And for the final time, the 4th and the final time, we will cover the renin-angiotensin system again. And then we move on to the reproductive system. So in lab, I suppose you all are working on like the male reproductive system. You all started female. Okay, this is working on that. Okay. So we will cover the physiology related to the male and the female reproductive system. And then from there, move on and talk about fertilization of sperm and the ovum and pregnancy, changes happening obviously in mom, as well as all the changes that need to happen developmentally. So prenatal development, right? Embryonic and fetal development. And then we will move on to talk about right after birth, all the changes that should happen there, lactation, oh, the labor process as well, labor delivery, all of that. So there's that part of it, and then a little mention on genetics. Okay, so I think most of the genetics topics you'll actually cover in lab. I will reinforce some things, and a few things that haven't been covered in lab is what I will talk about. Okay, so let's go ahead. This is so we got started with this, right? Just like right before you took your third exam. So water balance is something we definitely need to discuss because water is constantly shifting. This is just the process of diffusion or osmosis, which is the movement of water from a high concentration region to a low concentration region. This is happening as water shifts out, like from within the cell, there's an intracellular fluid compartment. So water shifts from within the cell to outside the cell where you have spaces between the cells. We call these interstitial spaces, or fluid there is interstitial fluid. And then specifically shifting water into the blood vessels where this becomes part of plasma, blood volume, right? So there's always going to be shifting of water between these three compartments. And I think we talked a little bit about this. Osmolality is really the concentration of solutes in the entire solution. The biggest factor that kind of guides ECF, a water balance in the extracellular compartment is really osmolality. So as you see an increase in osmolality, which means you're seeing an increase in solutes, and therefore correspondingly that should be that should be a reduction in the water content, right? So when you see this, as water content drops in the extracellular compartment, that is obviously in relation to the intracellular fluid compartment. And therefore, you're going to start seeing water shifting back and forth from high concentration to a low concentration area. So I'll go ahead and explain this one more time. The water balance is really controlled by how much water you are putting into your body. So intake versus how much are you eliminating from the body, mostly in terms of urine, so output, right? So I believe we covered all of this. Y'all can remind me. I think we did. Yeah, okay. Um, so when it comes to intake, all right, so the biggest force here would be, again, ECF osmolality. When you see an increase in ECF osmolality, which obviously then corresponds to a drop in blood volume, okay? Let me go ahead to this next slide and explain it. I'm pretty sure we've covered this too, but let's go ahead and do it again. When you see an increase in ECF osmolality, this tells me that the overall solute concentration has gone up, and therefore water concentration in the extracellular fluid compartment goes down. So obviously blood is part of that extracellular fluid compartment. So if you're saying water concentration is reduced in the extracellular compartment, that would then mean that your plasma volume is also dropping, right? I mean, this is both, they both kind of go hand in hand. So let's talk about this. When you see an increase in ECF osmolality, this information is sensed by chemoreceptors, basically, right? And this is by osmoreceptors within the hypothalamus region of the brain responds to this, and this activates the thirst center, and so allows you to consume more water. So you're drinking more water. That water is going to go through the entire GI tract, gets absorbed in the small intestine. It's absorbed within the villi where you've got blood vessels there, right? And as you absorb that water into the blood vessels, well, that's obviously going to correct for this problem, right? Therefore, putting more water back into the extracellular fluid compartment and recalibrating this problem right here, kind of balancing that out, right? But as you put more water into the blood, then obviously this also would correct for a drop in that plasma volume as well. So they fix both, right? But you also do know when you see a drop in plasma volume, then you also correspondingly would see a drop in blood pressure. And this is where the kidneys come into play, which we've talked about, is those granular or JG cells in the wall of the afferent arteriole would get activated in response to this drop in blood pressure. And when those granular cells get activated, this releases renin. Renin causes the conversion of the Ang1 to Ang2, which then activates aldosterone. Aldosterone is released from, y'all remember, the adrenal cortex, right? Aldosterone is going to get circulated through the blood, find receptors specifically in the distal convoluted tubule, right? When it binds that, then we know what happens in terms of what aldosterone does, right? Sodium reabsorption, water reabsorption, which then helps to, again, correct that problem in the ECF where water concentration had gone down. Angiotensin 2 by itself, okay, does a lot of different things. It is a vasoconstrictor. It then ties in with what the aldosterone mechanism needs to do next. So that's the other piece of it. But angiotensin 2 can also activate that thirst center in the hypothalamus. So there's actually like a feedback between these two components. This is input, right? So how do you regulate how much water you put into your body? Well, that kind of depends on osmolarity within the ECF compartment, tied in with what the hypothalamus needs to do, activating the thirst center, the renin-angiotensin mechanism, all of that works together. That's one way of controlling water balance in your body, right? How much you put into your body. But you can also control or regulate how much urine is eliminated from the body. So it's really a balance between intake and output, right? Okay. So when it comes to output, this is all controlled by ADH mechanism. So antidiuretic hormone, again, all in relation to the same issue where you see a drop, an increase in ECF osmolality. What's going to happen then is as the water concentration drops in the ECF compartment, you could also activate ADH. ADH is made, again, if you remember, back in the hypothalamus, and it's released from the posterior pituitary. The receptors for ADH, you all know, are, again, back in the kidney, renal tubule, but specifically in the collecting duct this time, right? And what happens then? Okay, I think this next slide talks about it. Here we go. When you see an increase in ECF osmolality or a drop in plasma volumes, this was the same thing that we talked about for input, but this is also now controlling output, but except the mechanisms are different. For input, We're talking about the thirst center within the hypothalamus, the renin, angiotensin, aldosterone pathway, that's input. The output, how do you control output, which is how do you control urine output? This is ADH, antidiuretic, right? So they kind of work hand in hand. So again, same thing, receptors that get activated in the hypothalamus, allowing what? The posterior pituitary to release the ADH. ADH is going to target those collecting ducts. What happens then? We open up aquaporin channels within the collecting duct, allowing sodium to be reabsorbed from the filtrate, where back into the blood, peritubular capillaries, right? And so as you put water back into the blood, obviously blood volume goes up, which then would correct that ECF osmolality issue that we started off with, right? Okay. Of course, if blood volume goes up, urine output goes down, and that's why this is antidiuretic, reduction in the urine output. Okay. So really, when you're looking at water regulation or controlling a balance of water between the different compartments, this is how do you regulate intake versus output? And you really need to talk about both, right? Because it's a balance between the two. Is that clear to you all? Okay. All right. Here's some of the disorders related to water balance. Okay, nothing kind of new here. Y'all have heard of a lot of these terms, right? But let's go ahead and put this in perspective in terms of like how is water shifting between the different compartments? And I'm mostly focusing on intracellular versus extracellular, right? So when you look at dehydration, this is overall water loss, but we're mostly looking at water loss in the ECF compartment, not necessarily within the cell. It's outside of the cell in the interstitial spaces, as well as if you see water loss within the blood. We're talking about a drop in blood volume, right? So anything when you're seeing a drop in water content within the extracellular compartment, this is what I call dehydration. A lot of different reasons why this might be happening, right? So really hot day, so sweating, so you're eliminating a lot of water in terms of perspiration. Of course, there could be other reasons too that could result in this overall drop. Hemorrhage could be a reason too. It's a hypovolemic shock. Okay. So I'll start here. And there's a few schematics that come up that actually explain this a little better. Hypotonic hydration. So this is kind of like an overhydration issue, right? The opposite of dehydration. This is when you consume too much water too quickly. All of that water obviously is absorbed in the blood. From there, it's going to shift out of the blood into the interstitial spaces. From there, it's going to shift into the cell, right? So then you see this. Okay, hold up. Okay. All right, then you kind of see water accumulating within the intracellular fluid compartment, and this is what I call overhydration, cellular overhydration, or what we call water intoxication. So again, as you see disturbances in water balance, that's going to also cause disturbances in sodium levels. And then if you think about it, that also causes disturbances in potassium. And I'm about to explain to you, when you see potassium changes, then that messes up hydrogen ion changes as well. So they kind of all link together. But the biggest thing that comes to mind is when you put too much water into the cells, you got to worry about what's happening. Are you diluting out your sodium content, right? And you do need sodium, you need potassium as well. You need both of those for what? Action potentials, right? I mean, action potentials of any excitable cell, skeletal muscles, neurons, cardiac muscle, all of that. So that's why we have to worry about water balance, because this really also changes ionic balance as well. What's edema? Okay, edema is basically, again, this is not dehydration, right? The opposite, right? This is an accumulation of fluid. But again, this is very specific to the interstitial spaces. So if you see a buildup of interstitial fluid, that's what we typically call edema. So interstitial spaces are the spaces surrounding the cells within the tissues, right? Not necessarily talking about blood here, right? Not plasma, okay? Okay, so let's talk a little bit about dehydration first, right? And then we'll talk about the opposite scenario. So when you're looking at dehydration, again, what is dehydration? This is loss of water or excessive loss of water from the extracellular fluid compartment. I've got cells here. So what you see inside the cell should be intracellular fluid. Everything outside here is extracellular, right? Okay. I'm saying I'm losing water content here, right? If I'm losing water content here, that would mean in comparison within the cell, it's all relative. Does that make sense to you? It's like you're always comparing what's happening inside the cell with what's happening outside the cell. So if outside the cell there's less water because dehydration, right? Then in comparison, that would mean you have more water content or more water concentration within the cell, right? So you're seeing a gradient here, a concentration gradient for water, a higher amount of water within the cell, less on the outside. So when you have a gradient for water, this is osmosis, right? So what's going to happen? What is going to shift across this plasma membrane of the cell, always moving from a high concentration to a low concentration. So what's going to happen? What is going to move out of the cell to compensate for this loss of water in the extracellular spaces? That makes sense, right? So yeah, so this is good. We do want that, right? We want that shifting to happen. But if you had extreme dehydration, then what would happen? You would see a lot of that water shifting out of the cell to correct that problem. And then you would have shrinking. That's not good. Shrinking of the cell, and that's not good for cellular processes within the cell as well, right? It's a normal mechanism. We want that to happen, but not in an extreme kind of a situation, right? Okay. On the other hand, if you see right the opposite issue, this is hypotonic hydration. What's happening here? This is where you're consuming too much water. It's obviously being absorbed into the extracellular, into the blood first, right? Which is part of the extracellular space. You have way too much water here, right? In relation to what you're seeing within the cell. So how would water move? Always from high to low concentration. Because you have a lot of water, you consume too much water too fast, right? Overhydration. What's going to happen? That water is going to shift from the blood to the interstitial spaces and then from there into the cell. And as that happens, you see swelling. So again, that's not good too, because too much swelling could result in rupture of the cells, which is definitely not good. That makes sense, Joe. So when you have dehydration and overhydration, on the other hand, you're seeing water constantly shifting, and this could affect how the cell functions because too much water leaving the cell causes drastic shrinking. We don't want that. Too much water entering the cell causes too much swelling. Don't want that too, right? So it's always a balance, but you got to keep that in mind when it comes to water balance. And this is why the hypothalamus and the whole renin, angiotensin, aldosterone mechanism, along with when ADH needs to kick in, all of this is super important because it keeps this balance, right? Okay. Okay, so then when you're looking at water balance, the biggest ion that contributes to water shifting back and forth is really sodium. Okay, with the kidney, we talked about those juxtamedullary nephrons, which 15, 20% of the nephrons, right? Where you had this really long nephron loop that extended into the medulla. And as the filtrate moved down the descending limb, you remember how we concentrated that filtrate, right? Remember this, because the descending limb is selectively permeable. It only allows water to leave, but not the salt. As a result, you're really concentrating the filtrate. We went for it from 300 milliosmolar to the deepest point, about 1200 or something like that, right? And then as we started moving that filtrate back up the ascending limb, right, the opposite happened because the ascending limb is selectively permeable only for salt, but not water, right? And as this concentrated filtrate started moving up the ascending limb, now you're pulling out solutes like sodium and potassium and all of that. But you're not touching the water, so you started to dilute that filtrate back up, right? This is what was used. This is what we call the medullary osmotic gradient. And this was extremely important for the collecting duct then to be able to either pull, reabsorb more water if needed in the case of dehydration. So what the kidney does is super important to kind of maintain that how much urine is being eliminated, which ties in with blood volume and of course blood pressure, right? But as you talk about ADH mechanisms and reabsorption of water, you also, and then aldosterone also plays a role with the sodium, water reabsorption, but it starts with sodium first. Really what contributes to this osmolality, which is again, any fluid within the body, I mean, you saw that it got concentrated, but then ultimately any fluid in the body, biological fluid, has to be maintained at a specific kind of an expected range of osmolality, somewhere between 280 to about 300 milliosmolar. And a lot of that, like again, remember when you think about osmolality, it's a concentration of solutes in relation to the entire solution, right? And the solution is solute, but also solvent. What is the solvent here? Water. Right. So if you're talking about the water concentration in relation to the solute concentration, that's what really determines what that osmolality ends up being. And the biggest solute that helps with that is sodium. And that's why we need to talk about that, because whatever sodium does, kind of water follows right behind it. So we have to control sodium levels, right? So, and the mechanisms that do this, the biggest mechanism, hormonal, would be the aldosterone mechanism. Okay, so let's go ahead and talk about this. Yep, okay. What slides? This is just explaining what the mechanism of renin, angiotensin 2, aldosterone was. So we know a lot of this. How do you control for sodium levels within the kidney? Uh, you know that sodium was filtered out of the glomerulus. It left the glomerulus, it became part of the filtrate, it and went through the entire renal tubule, different regions of the renal tubule. The first part is the proximal convoluted tubule. You all know that this is where about 65% of reabsorption is occurring. It is not regulated by hormones, not in that region, correct? So about 65% of sodium gets reabsorbed along with water and a lot of other good things, right? Glucose and all that. Uh, so that's where you're seeing the majority of that sodium being reabsorbed from the filtrate. You do not want to eliminate sodium in urine, right? I mean, you want that reabsorbed back into the blood because those sodium levels are important for maintaining osmolality of body fluids, right? Okay. We also know that about 20-25% or something of the sodium gets reclaimed in that nephron loop, but specifically not descending the ascending limb, correct? And then a regulated control or reabsorption of sodium is occurring in the DCT along with the whole aldosterone mechanism. So I think there is a slide that puts everything together. This is just talking to you about the aldosterone angiotensin mechanism, nothing new here. You all know the biggest trigger here is a drop in blood pressure. That's what's going to cause renin release from the granular cells. And that ties in with the Ang2 mechanism, which is a vasoconstrictor, but that ties in with aldosterone as well. What slides? I think it comes up here and then a few other slides too. I'm just going to focus on the schematics because that puts together all of these concepts. Okay, so everything that I'm talking about right now, I mean, I hope that you kind of remember these details because nothing new so far, okay? Only when I get to acid-base balance, you'll hear some new content. Okay, we do know the adrenal cortex is what's going to release the aldosterone. The aldosterone, being a hormone, will circulate through the blood and then target the DCT within the kidney tubule, which allows for two things to happen because aldosterone really controls two things. Sodium gets reabsorbed. And what do I mean by this? Reabsorption of the sodium from the filtrate, correct? But you're reabsorbing it back into the blood, correct? But at the same time, potassium can also get secreted. Now, this depends on whether we have excess potassium within the blood. And when you do, if you do have that situation, then excess potassium could get secreted back into the filtrate so that it can be eliminated in urine, okay? Okay, when you see sodium getting reabsorbed back into the blood, what follows right behind it is water, right? So as a result, you are putting water back into the blood as well. Therefore, blood volume goes up, blood pressure goes up. But because you're pulling that water out of the filtrate, urine output goes down, okay? Okay. How else can you control sodium levels? If it's tied to aldosterone, well, ANP, atrial natriuretic peptide, is an antagonist for aldosterone. So again, this is kind of tied in with blood pressure regulation. When you have extreme high blood pressure, in this case, that's when ANP is released. And ANP is going to be an antagonist to aldosterone. It's going to shut down that whole pathway, right? The renin angiotensin pathway. So here we go. This is explaining ANP mechanisms. So when you see an increase in blood pressure, which obviously increase in blood pressure would mean stretch to the blood vessel wall as well. So that's another way to think about it. This is when ANP is released from the heart. What does this do? Well, one is, again, we're going to actually kind of silence those granular cells at this point, right? Because granular cells get activated when the blood pressure is low, right? We're just going to inhibit that. So less renin is being released, less angiotensin 2. Angiotensin 2 is a vasoconstrictor. If you lower Ang2 levels, what's going to happen? Vasodilation, right? And if it's vasodilation, you're opening up the blood vessel. What happens to resistance? Resistance drops. And if resistance drops, blood pressure drops, right? So the purpose of ANP is really to control blood pressure. So when blood pressure is too high, ANP is released. And its main goal is to correct high blood pressure by lowering blood pressure. One way to do this is to silence that whole renin angiotensin mechanism, right? I mean, just downregulate it. Okay, what else? Um, ANP can also feed back. This information can also feed back to the hypothalamus and the posterior pituitary, where now you are releasing less ADH. So this is right the opposite of what we were talking about with aldosterone mechanism, right? Because again, the problem here is right the opposite. It's not low blood pressure, it's high blood pressure, right? So when you release, I'm sorry, when you inhibit ADH, what happens? There's less of that water being reabsorbed. And as a result, there's blood volume goes down, and that's going to reduce your blood pressure. Okay. And notice ANP is also going to be an antagonist for aldosterone, which ties into the same mechanism here, right? So if you're looking at this, ANP is antagonizing three different things, really. It's antagonizing renin release, which shuts down renin Ang2 aldosterone. By itself, it reduces the release of ADH from the posterior pituitary, and it also antagonizes the release of aldosterone from the adrenal cortex. So it's all right the opposite of what we've been talking about. Okay. Okay. But you understand why we need to talk about ANP, because um, aldosterone, when it accomplishes its goal, what happens then is that blood pressure becomes much higher. And so we need kind of a balance there, right? We need to maintain homeostasis. This is where ANP kicks in to kind of negate some of the effects of aldosterone till we find a balance, homeostatic balance. Okay. Okay. Here is my final summary slide. Okay, so this is really a summary of a lot of topics we've been talking about throughout the semester. So this goes back to cardiovascular. This goes back to how you're controlling blood volume and blood pressure, but it ties in cardiovascular with urinary, right? So what the kidneys have to do. But this also ties in endocrine, and it ties in neural mechanisms like sympathetic activation. It ties in everything together, right? And so let's look at this. Again, nothing new here, but let's break this down. So there's two ways you can think about it. The biggest trigger, though, is when you have a dramatic drop in blood pressure. So, okay, I want to point out, this is not that, remember how we talked about what the kidneys do, intrinsic mechanisms versus extrinsic mechanisms. Intrinsic mechanisms is if you see a slight drop in blood pressure or a slight increase in blood pressure, but you're still operating within your MAP range, 80 to 180, then it's your intrinsic mechanisms, your myogenic response, and then how those macular denser cells feed back, right? Remember all, okay, we're not talking that, right? Here we're talking big picture, systemically we got a problem, blood pressure is too low or too high, right? And what this slide is talking about is what happens when the blood pressure is too low, right? You can reverse all of this explanation if you were looking at an increase in blood pressure. But so this is your systemic response. This is your extrinsic mechanisms where the kidneys are not that concerned about maintaining a fairly constant GFR. Remember that, right? That's not the goal here. Here, the goal is we've got a dramatic drop in blood pressure. How do we correct it? Okay, all hands on deck. We've got neural mechanisms, we've got hormonal mechanisms, we got the kidneys, we got everybody intervening to correct this problem, right? So this is the big summary of a lot of different concepts we've been discussing. throughout the semester. Cardiovascular, renal, neural, hormonal, everything. So let's look at this. Okay. What happens when you see a dramatic drop in blood pressure? You know that blood volume, blood pressure kind of ties together, right? So drop in blood pressure, drop in blood volume, same thing. All right. What happens? So this is my problem. What should I do? I want to correct it by increasing blood pressure. You see that, right? This is the issue, and this is negative feedback mechanism. I have to correct it. How can I correct it? Four different pathways. Do you see this, right? So what's the biggest, what is the biggest uh, pathway? Well, not biggest, the fastest responding pathway. When your blood pressure drops really dramatically, this is where we're activating fight or flight response, right? So we're talking autonomic nervous system, sympathetic response. We're going to activate the sympathetic nervous system. So what is this? Do y'all remember? This is where sympathetic neurons are activating cells within the adrenal medulla. Epinephrine and norepinephrine is released. This goes directly into the bloodstream, right? You remember this, right? And then what does this do? A lot of different things, but one of the things it does is it targets the blood vessels, bringing about vasoconstriction, right? Vasoconstriction means narrowing of the blood vessel diameter. What does that do to resistance to blood flow? Goes up. And if resistance goes up, what happens to blood pressure? Goes up. You see that? So this is my first pathway. Active, this is neural, right? This is through the sympathetic response. I am increasing peripheral resistance, which then directly ties to blood pressure increase. Do you see that this pathway has nothing to do with blood volume, right? This goes directly vasoconstriction of the blood vessels, which increases your blood pressure. Second mechanism is what the kidneys are doing right here. Okay, what's going on here? When you see a drop in blood pressure, this is kind of, again, related to that feedback mechanism or what those macular denser cells are doing. But really what you're seeing here is reduced stretch. So therefore, you are activating that renin pathway. Angiotensin 1 gets converted to angiotensin 2. You know the rest of the story. Aldosterone gets released from the adrenal cortex. When aldosterone gets released, this is going to bring about increased sodium and water reabsorption. You're reabsorbing the water into the blood, therefore blood volume goes up. Likewise, urine output goes down, right? So if blood volume goes up, then blood pressure goes up. So you notice that this is the hormonal pathway, right? And this actually first increases blood volume before it brings about an increase in blood pressure, okay? The other mechanism, Ang2 by itself, I've always said this, is also a vasoconstrictor. You see how systemically through the sympathetic nervous system, we're bringing about vasoconstriction, right? Okay, but Ang2 is also a vasoconstrictor. So we're just complementing each other, right? We're just augmenting the response. So again, vasoconstriction means increase in peripheral resistance, which will increase your blood pressure. So neural hormonal mechanism in relation to the kidney. And the last piece is this, right? Okay, just as much as the aldosterone pathway kicks off, so does ADH. So when you see a drop in blood pressure, the posterior pituitary is going to release all that stored ADH. ADH does what? Targets the collecting ducts. No, nothing about sodium reabsorption here. This is directly water reabsorption. And water reabsorption means putting that water back into the blood. So blood volume goes up. Of course, urine output always goes down. And so blood volume goes up, blood pressure goes up. So this is really a summary. This is the big picture slide, right? This is showing you how do you regulate water, right? Which kind of ties in with how you're regulating sodium. And all of this is important for controlling blood volume as well as blood pressure. Does that make sense to y'all? Yes, ma'am. Oh, all together, all working together. Okay, how fast though? Neural, very, very quick, right? We're talking action potentials, electrical signals, very, very quick. Once the epinephrine and norepinephrine is released, direct access to bloodstream. So we're going to go get our targets pretty quickly, right? Within a matter of like minutes, that could happen. Hormonal release, we got a few things that need to happen. Renin has to be released from the kidney. Angiotensin 1 to angiotensin 2 conversion needs to occur. That has to then cause the release of aldosterone from the adrenal gland, which the, no, yeah, cortex. And then from there has to come back to the kidney to bind receptors in the DCT, then bring about sodium and water reabsorption. So obviously a little bit longer than a few minutes. Does that make sense? Hormonal is always going to be uh takes a little longer because there's a lot of things that need to happen different places, but it's not as fast acting, but tends to be a longer duration response. Yes, ma'am. Yes, I would say to a certain extent. It just depends on what kind. Okay, so how fast do you want it to compensate? Is that blood pressure tanking big time? Okay, so then if you do not have the kidneys helping out with the situation quickly because there is kidney dysfunction or something like that, then yes, you're going to put more pressure on the neural compartment, right? But we want everything to work together. Yeah. Okay. So I think, yes, we've talked about everything related to sodium, which obviously ties in with water balance. Last piece is I want to quickly talk about potassium. So, okay, uh, this is really controlled by aldosterone. So if you had excess potassium, well, let's talk about this. Why do you need potassium? Why is this so important within the body? Well, very important for all of your excitable cells, part of the action potential. You need sodium for the depolarization phase of the action potential, but to repolarize the cell, you do need potassium, right? And again, if you have an imbalance with that potassium, this could result in cells staying excitable for a longer period of time, or may not be able to get it to that excitation state. It again, it's a balance though between both sodium and potassium. So what happens if you see an increase in ECF potassium levels? This is hyperkalemia, and that's going to basically decrease your resting membrane potential. So again, you're not going to be able to get those cells, those neurons and cardiac and skeletal muscle cells excitable, right? Right, the opposite, if you have less levels of potassium in the ECF, this is hypokalemia, okay? Right, the opposite, hyperpolarization in this case. So I want you to think about this also in relation to hydrogen ions. Okay, that looks okay. It looks better. It looks better on your recorded video, but okay. I've not had the chance to look into the conversion here, but okay, work with me. I have the intracellular compartment here on the bottom and the extracellular compartment there on the outside, right? So if I'm looking at hyperkalemia, which means an excess potassium here in the outside in the extracellular compartment, right? Here's what's going to happen. As you build up potassium ions in the extracellular space to compensate for that, potassium is a positively charged ion, right? To compensate for an increase in positive ions in the extracellular space, hydrogen ions will shift into the intracellular space. Makes sense? To kind of balance out your cation concentration. So this is one of the things we need to worry about in the case of potassium imbalances. As you see hyperkalemia, hydrogen ions are going to shift inside the cell, and as hydrogen ion concentration increases, pH drops, and so you start to experience acidosis. So that's one thing we've got to keep in mind, right? Right, the opposite is going to happen if you have less potassium ions on the outside, like hypokalemia. That's going to result in less hydrogen ions on the inside of the cell, which would mean pH goes up. This is alkalosis, right? So this kind of ties in with pH balance. Okay, how do you control potassium levels? This is controlled through aldosterone, which means DCT is the site, distal convoluted tubule is the site where those receptors would bind aldosterone. It does two things, sodium reabsorption, but also potassium secretion. Okay, calcium. Back to the DCT again, where you have receptors for aldosterone, you also have receptors for parathyroid hormone, PTH. What does that do? Okay, we know that. Okay, typically when you start to see low blood calcium levels, this is hypocalcemia, This is what's going to prompt the release of PTH. A lot of this was discussed in ANP1, okay, when we discussed bones. Okay, when PTH is released, and also in the endocrine chapter earlier on this semester, right? PTH gets released from the parathyroid gland. It does three different things, but the biggest thing it does is this, is PTH is going to target those osteoclast cells. This is a population of cells within the bone tissue, right? What do osteoclast cells do? They resorb or break down bone matrix. And what's stored in that bone matrix? Calcium, right? So as you break down strategically certain areas of osteoid, you're also going to release stored calcium from those regions. We're going to release it as a soluble form. You're going to put it back into the blood. Therefore, you're correcting this hypocalcemia problem. We're increasing blood calcium levels. You see that? That's one way to do it. Okay. If you want to put back more calcium into the blood, then you would also need to kind of basically calcium reabsorption could also occur in the distal convoluted tibial. This could also be vitamin D activation, which allows for more calcium to be absorbed in the small intestine from the food that you are eating, consuming. Okay. All of these three mechanisms are going to increase calcium levels in the blood, correcting hypocalcemia. Okay. Okay. Ooh, sorry. Okay, so we've talked about water. We've talked about how sodium plays a huge role in that water balance. We also talked about potassium and hydrogen to a certain extent, and then calcium, right? Last piece is focusing on hydrogen ion concentration because this ties in with acid-base balance. So if you're looking at, okay, nothing, the first part is not new. I hope you can remember some of the details we covered in ANP1. So when you're looking at pH, y'all should remember that pH is a scale that goes from 0 to 14, 7 being right in the middle, neutral. So anything 6.99 all the way down to 0. So anything below 7, right, is considered acidic. As pH drops, what happens to hydrogen ion concentration? Goes up, right? Okay. What about 7.01 all the way to 14? All of that is alkaline or basic territory, right? And as we go up the pH scale, hydrogen ion concentration goes down. It's an inverse relationship. Okay, so we got to maintain our pH at a fairly constant level. So pH of blood is, generally speaking, 7.35 to 7.45. That's my range. It's going to change a little bit between what you see in the arteries versus the veins and so on and so forth, just a little bit. But overall, you would say pH has to be maintained. It's a very tight range. That's like a 0.1 difference, right? 7.35 to 7.45. So what happens when pH drops? We said this is going to result in acidosis. So do y'all remember how do you correct acidosis? If you have a chemical buffer, which consists of a weak acid and a weak base, you correct acidosis with the help of a base. Okay. We talked about the calcium, we talked about the bicarbonate buffering system, right? The weak base there would have been bicarbonate. So the bicarbonate is able to bind all of this excess hydrogen ions and therefore kind of move that pH a little higher to bring you back within this operating range. The opposite is true if pH increases. So this results in alkalosis. As pH increases and alkalosis occurs, at this point, I don't need that base. I need the acid instead. I need the weak acid. And the weak acid that we used in that bicarbonate system was something called carbonic acid, H2CO3. All right, so what does the carbonic acid do? In the case of alkalosis, when you recruit the carbonic acid, it's going to dissociate. It's going to release those hydrogen ions. And as you release hydrogen ions, you're going to drop the pH. And so you were going this way towards the right. Now you're going to drop it and hopefully get you back into that operating range, right? That's how a chemical buffering system works. And we talked about the chemical buffering in ANP1 because it's the fastest acting system to control or to regulate pH levels. Can happen within a matter of seconds to a few minutes, right? I mean, you can, when you see changes in pH, you can quickly, if the pH is dropping, we can quickly increase the pH with the help of a base. If the pH is going too high, we can drop that pH with the help of an acid very quickly, right? Okay, So we talked about this. All right. So there are different reasons why this pH might fluctuate within the body. So when you think about acidosis, what are some of the reasons why acidosis could occur? Meaning we're building up acid. Acids of different types. A lot of it is metabolic driven, really. So y'all know when you metabolize glucose, you release two pyruvic acid molecules. This is glycolysis. Then that what happened to the pyruvic acid? It got converted to something called acetyl-CoA that allowed you to bring it into the mitochondria to go through the Krebs cycle, right? Okay, but I need oxygen for that to occur. In the absence of oxygen, or if you have low levels of oxygen, the pyruvic acid starts kind of building up, which is not good. This immediately gets converted to lactic acid. Lactic acid, another acid, right? So that's going to drop pH. That's one reason why you could be seeing acidosis. So this could be happening like you could be seeing imbalances like this happening every day, right? As part of the normal metabolic process, depending on the availability of oxygen and how fast you can get that acetyl-CoA into the citric acid cycle. Okay, as you metabolize lipids instead of glucose, instead of carbohydrate metabolism, as you metabolize lipids, so what are we talking about? Fatty acids, glycerol. You all remember how fatty acids can get directly converted to what? Acetyl-CoA, that process called beta oxidation. You can also tap into the glycerol, convert it to G3P, glyceraldehyde 3-phosphate, and then utilize that. But do you all remember, again, back to acetyl-CoA, if acetyl-CoA keeps building up, right, and I'm not able to pull it into the citric acid cycle because you need a pickup molecule there, if oxaloacetic acid, exactly. If that acetyl-CoA builds up because you're metabolizing lipids too fast, what happens? We take it to the liver. And then what happens to all of that? Gets converted to something called ketones or ketone bodies, which is also an acid. So that's going to drop the pH, acidosis. Here we go again, right? So that could cause a problem there as well. Okay. As you break down proteins, which doesn't happen very often, there are some organs it does happen, like the liver. But if you need to break down proteins, then you're probably releasing things like phosphoric acid as well. Again, another source for acidosis. Also CO2. This is respiratory control mechanisms. If you have an imbalance, hyperventilation, hypoventilation, any kind of ventilation imbalances related to the respiratory system, what happens then? If CO2 starts building up, so this is hypoventilation, right? I mean, that's when you start trapping CO2 within the alveoli, the blood, and so on and so forth. What happens when you start trapping CO2 within the blood cells? pH is gonna go down, right? Because it's an acid resulting in acidosis. So a lot of these reasons could result in acidosis, and this is why you need to have corrective mechanisms. What are your corrective mechanisms? Okay, here are your three big corrective mechanisms. Chemical buffering, there's different chemical buffers. The biggest extracellular chemical buffer is the bicarbonate system, which we covered in ANP1. Today, I will talk to you about what's going on specifically in the kidney, where they use a different chemical buffer called the phosphate buffering system. So we'll cover that. Earlier in the semester, we did talk about the respiratory control, CO2, again, hyperventilation versus hypoventilation. And that's super important for controlling acidosis and alkalosis. And again, for today, I will focus on renal mechanisms. So now that we've covered the urinary chapter and you're familiar with the different regions of the tibial, let's talk a little bit about how you control hydrogen ions and bicarbonate ions, right, to control your pH levels. All right, so the fastest acting system is your chemical buffering within a few seconds to a minute or so. Respiratory mechanisms pretty quick too, within a few minutes. Renal mechanisms take a little longer, normally a few hours or so before you really see any appreciable change. But again, if you're looking at long-term control of pH, it's renal. It's what the kidneys are doing. If you want quick changes, it's chemical buffering, and certainly what the lungs are doing in controlling CO2 levels. So when you're looking at renal regulation, all right, the next few slides can look a little scary. There's a lot of schematics that kind of, this is all new content. You've probably not heard about this before. So I'm going to keep it very simple. Okay. I just want you to follow two important ions. We're going to focus on what's going on with those hydrogen ions, right? Everybody clear? If hydrogen ions builds up, what does that do to pH? Go down, right? Go down. And what is that condition called? Acidosis. We don't want that. Okay, we got to control those hydrogen ions. And then bicarbonate is part of that weak base, right? Which is very important to control that acidosis issue. So I'm going to talk about these two important ions. And of course, the context here is kidney. Ions come through the filtrate as it goes through the PCT and the nephron loop and the DCT and collecting duct, right? We're going to talk about these different regions and how you control for hydrogen ions and bicarbonate. Okay. All right. One more thing. Let's go ahead and draw this out. This is a reference. I'm not going to do a whole lot of this today because a lot of this is on my slides. Blood. Okay. That's fine. And filtrate. And this is the kidney, tibial, different regions of the kidney tibial. We'll break this down. And obviously those would be your peritibular capillaries, where you see back, right? Okay, so what do I mean by reabsorption? We're starting from here and we're reabsorbing into the blood. Is that right? Okay. That's reabsorption. Okay, what do I mean by secretion? Right, the opposite. We're pulling something out of the blood, moving it there, correct? That's secretion. Keep that in mind. We're going to add some details, and that's what we're focusing on, okay? So when you are looking at how the kidneys play a role in controlling pH, again, most of what the kidneys are doing is it's eliminating what we call fixed acids or non-volatile acids. A lot of this is that phosphoric, uric acid, lactic acids, ketones that are building up, all of that, okay? Again, we're thinking long-term control. A lot of this is going to take some time, okay? The way this works is, okay, so several mechanisms, and I'm going to work through all of these mechanisms. Okay. Let's have this here right now. Two things I want to keep in mind. I want to be able to reabsorb HCO3 minus. What's that? Bicarbonate. Do you see that? That's my weak base. I want to be able to reabsorb that back in the blood because this plays a huge role in the extracellular buffering to maintain, to correct acidosis. I want to make sure I conserve all of that. If I don't do anything with that bicarbonate, you know it's going to go through that entire filtrate, through the entire tubule, and you would eliminate it in urine. I don't want that. I want that bicarbonate to be reabsorbed into blood, okay? But for every bicarbonate I reabsorb in the blood, I want hydrogen ions to be secreted back into the filtrate. Does that make sense to you? Because if this builds up in the blood, what happens? Acidosis, right? Don't want that. I want to control those hydrogen ions. So every bicarbonate that moves into blood, reabsorbed into blood, I want to secrete hydrogen ion back into the filtrate, where I'm going to do something with it. I'm going to explain it to you. Does that make sense? It's just that. It's as simple as that. A bicarbonate is getting reabsorbed into blood. Hydrogen ions are getting secreted back into the filtrate. So let's talk about this. Yes, a lot of steps, really, if you, okay. This is what I have on the board. I'm going to show it to you here with my schematic. So this is happening in the proximal convoluted tibule, which is the very first part of that renal tubule. You have your PCT cells right here, right? And on the left, this is the filtrate coming through the proximal convoluted tubule. So I'm going to talk about movement, got to move things from the filtrate through the cell into this blue space, which is outside the cell, interstitial spaces, right? Then from there into this blood vessel, which would be your peritubular capillary. Do you all see that? So what is reabsorption? If I pull anything out of the filtrate and I put it back into the blood, that's reabsorption. If I pull something out of the blood through these cells and put it back into the filtrate, what is that? That's secretion. We got this? Okay, here's what, lots of different pumps here. All right, I'm going to try to keep this simple. Okay, again, this kind of the same legend that you've seen in the previous chapter as well. Primary active transport, solid arrows, secondary mechanisms, co-transport mechanisms, dotted arrows, so on. And you have your ATPase pump here. So it really begins like this. Okay, so we are pulling CO2 and water, right, into the PCT, into the cell, the proximal convoluted tubule cell. Okay, you remember this? Water and carbon dioxide forms, these are all reversible reactions, so it can go both ways, right? These two form H2CO3. This is carbonic acid. This is actually your weak acid in your bicarbonate buffering system. When H2CO3 can then dissociate into hydrogen ions and HCO3 minus, this is bicarbonate. Bicarbonate is your weak base within that same buffering system. So it goes both ways. All right, so what happens within the PCT is the water and the CO2 combine to form the carbonic acid, which immediately dissociates into what? Hydrogen ions and bicarbonate ions. You see that? What do I want to do with the bicarbonate ions? That's good stuff, right? I mean, I do not want to lose those bicarbonate ions. What happens to the bicarbonate ions? I'm going to reabsorb it into the blood. Do you see that? Okay, how so? Well, this is different mechanisms. So this could be either these are all co-transport mechanisms. As sodium ions leave the cell, I might be pulling the bicarbonate ions along with me, right? Or that's what I call a symport mechanism. What is antiport? If the two ions are moving in opposite directions, right? So for example, as chloride ions are coming into the cell, bicarbonate ions could be moving out of the cell. So this is an antiport mechanism. What you see here on the bottom is a symport mechanism. Both examples of co-transporters, right? Using both these mechanisms, the bicarbonate is being reabsorbed out of the cell, tubule cell, into the peritubular capillary. It's being pulled back into blood. That's really what I want you to see, is the bicarbonate is being reabsorbed into the peritubular capillary, into that blood, right? What's going to happen to the fate of these hydrogen ions? Do I want these hydrogen ions in the blood? Nope, because that's going to mess up blood pH. We don't want that. What do I do with the hydrogen ion? I'm secreting it back into the filtrate. You see that? It's going the other direction. All right. When I secrete it back into, okay, when I secrete it back into the filtrate, it basically takes this and it goes back in reverse. So it's just that reaction going forward, backward, forward, backward. That's it. So what happens? Hydrogen ions in the filtrate combined with the bicarbonate. This is how I'm pulling the bicarbonate out of the filtrate, taking it through the cell and then out into the peritubular capillary, right? That's how I'm recycling these hydrogen ions. I'm putting it into the filtrate only for the purpose of grabbing those bicarbonate ions out of the filtrate so that I can bring it back in here and then redirect it into the peritubular capillary. So I'm using hydrogen ions right now because I am in the first part of the renal tubule, right? I'm able to utilize it. So you see that this is a feed forward mechanism. This is going to keep happening. For every bicarbonate ion I reabsorb into the blood, a hydrogen ion gets secreted back into the filtrate. You see that, right? And this is going to keep happening because at this point, I'm not too concerned about the pH of that filtrate because I'm utilizing those hydrogen ions for a good thing, right? I'm able to utilize those hydrogen ions to pull the bicarbonate, which I really do want, okay? So as long as so again, this is happening in the PCT because there's still a lot of room for that filtrate to go through, right? So that we have more areas to control those hydrogen ions. So, okay, that's happening in the PCT. Now, there could also be new bicarbonate ions being generated all because of metabolic processes, and that's what's explained in this next slide. So, for example, what do I have here? Oh, no, I'll come back to this at the end. This is the final step. I want to show you this. Okay, as you remember how you can utilize amino acids converted to keto acids, and then I can utilize that for metabolic needs for ATP. Okay, so glutamine is an amino acid that could be used for this process. Notice, okay, don't worry about the details. I'll go ahead and explain it. But as long as you understand that, I'm good. Okay, this is really how the kidneys are using different segments of the renal tubule to control for hydrogen ions and acid-base balance. So what happens to the glutamine? It splits into ammonium and bicarbonate, right? Okay, no brainer. What do we do with the bicarbonate? Very simple. Take that bicarbonate, reabsorb it into blood, right? So this is actually new bicarbonate that was brought about from that amino acid. Okay, it's not being recycled, it's new. Okay, but what do I do with the ammonium? Well, the ammonium, you see how the nitrogen is bound to the hydrogen there? That's good. That would allow, so that means, remember, free hydrogen ions really lower the pH. But if it's bound to something else, like if it's bound to, like in water, or when it's bound with the carbonic acid, it does not change the pH. So that's actually good. That's one way to eliminate these hydrogen ions. I'm just going to move it back into the filtrate and eliminate that in urine. So I'm eliminating excess hydrogen ions this way because it's bound to the nitrogen as part of ammonium. That's one way to, but while doing that, I'm controlling hydrogen ions. I'm controlling pH, but I'm also taking that bicarbonate because I need that. I need to put that back into the blood. Do you see that? Okay, so that's good. All right, so I'm going to back up a slide because I skipped this. This is happening where? This is happening in the tubule as well, but notice this is in the collecting duct. This is at that final stage, right? I mean, this is where urine is being formed, and now we've really got to control our hydrogen ions. So as we go through the PCT and as we go through the rest of the tubule, I'm going to utilize the hydrogen ions to make sure that we're recycling and reabsorbing the bicarbonate ions. That's the main purpose. When we get to the end, though, when we get to the collecting duct, this is where, notice what happens. Okay, same thing. What do I do with the bicarbonate? I always am going to reabsorb it into blood, correct? What do I do with this hydrogen at this point? Well, I'm at the end of the tubule, right? I'm at the collecting duct. This is where the chemical buffering system of the kidney comes into play, which is not the bicarbonate system, it's the phosphate buffering system. So what happens? I have a hydrogen phosphate that binds the hydrogen ion, forming dihydrogen phosphate. So I'm pulling all of this excess hydrogen at this point, coupling it with the buffer here, right? And eliminating it as dihydrogen phosphate in urine. So this is one way, this is how the kidneys are controlling pH. So I'm going to utilize the hydrogen ions if I'm in the early part of the tubule, only because the purpose of those hydrogen ions is every hydrogen ion that goes that way, I'm able to reabsorb bicarbonate back into the blood. That's very useful. I want that. But when I get to the end, what do I do with all the excess hydrogen if it was building up? Phosphate buffering system should take care of that. Does that make sense to you? Okay. All right. This might be an area you'll want to kind of look at a little bit, digest it, and if we need to go over it again, we will. There's a lot of information. Really, if you can understand what I have there, I'm good. Okay, but we can come back and look at this one more time if you need to. Okay, that should wrap up this chapter. And why won't this work? Okay, I'm going to go ahead and kind of look at the male reproductive system. I think the first part is, wait, where am I? Is just the anatomy piece. I want to go over some of those concepts. You should have reviewed this in lab, but I want to go over some of it. I'm sharing the male reproductive system slides. Do you all see that? Wait, I lost you. Okay, here we go. Do you all see it? Remote users? Anybody? Yes, perfect. Thank you. Okay, so I don't know, the first few slides is all about the anatomy portion. So I'm going to go ahead and explain some parts of it. Hopefully this is not new material. You should have covered this in lab, okay? So when we're looking at the sex organs or the gonads for the male reproductive system, it's the testes. When we get to the female reproductive system, we'll start talking about the ovaries, right? So the testes and the ovaries, they produce what we call sex cells or gametes. sperm in the case of the testes, and different things. It's called different things, but ultimately the ovum in the case of the female, the ovaries. Okay. All of these processes of forming sperm or forming the ovum, ova, it's guided by hormones, right? So a lot of different hormones, a lot of endocrine references back again here. Okay. So the biggest hormone that controls sperm production is testosterone. It's an androgen. Females, we're talking estrogen, progesterone, and a host of other different things, which we will talk about. Okay. There are always going to be accessory organs that help this, well, with the overall anatomy piece, which is going to guide sperm production or ovum production, right? Or follicle development and all of that. So we're going to talk about different glands and so on and so forth. So when you're looking at the male reproductive system, we'll cover the testes. What's happening within the testes is where the sperm is produced within the sperm factories called the seminiferous tubules. Then there's another region right outside. I'll use my schematics in just a minute, called the epididymis. So we'll cover all of that. There are three different accessory glands we need to talk about: seminal glands, the prostate, and the bulbourethral glands. Okay, so when you're looking at the pelvis, male pelvis, a lateral view of it, but also an interior view of it, right? So we're going to start here. You have a pair of male gonads, the testes, which you see here in white. It is housed within an external sac-like structure called the scrotum, okay? So you obviously notice that the male, the testes is not housed within the core of the male body. It is outside. I'll explain. But when you look at the cross section of this testes on the inside, there are 250, 300, some more than that probably, tubules called seminiferous tubules. And it's within those tubules that sperm are produced, okay? Through a process called spermatogenesis, which does not happen overnight. It's pretty spectacular. When you think about it, takes about, well, I want to say 60 to 72 days or something like that. So it takes quite a while to go from a stem cell to an actual spermatid, which then has to go through even more of a transformation to a mature sperm cell, right? So it is a pretty long process. Then once the sperm are produced, right, then they make its way into this next region here, which is called the epididymis, right? There's a head region, a body region, and then a tail, okay? And within the body of the epididymis, the sperm can then be stored. So during ejaculation, the sperm, it's not like the sperm was just produced and that's what you see as part of the ejaculate. No, the sperm were produced like almost 2.5 months ago and possibly stored in that epididymis for, depending on fertility and the age of the male, possibly 2 months. Okay, so sperm is produced here within the seminiferous tubules, can be stored in the epididymis, but when it is released, it's going to make its way through this tube called the vas deferens or the ductus deferens, which you see kind of loops around this structure here, the urinary bladder, right? It forms a larger kind of a region there called the ampulla, and that then the contents of the sperm in testicular fluid is basically then going to merge with the contents of this. This is the first accessory gland called the seminal gland. And the two together merge into a very short tube or a duct there called the ejaculatory duct. This orangish kind of structure that you see underneath that, that is the prostate gland. So I've got the ejaculatory duct, which is really the combination of what? The contents of the testes with the sperm, along with that seminal fluid right there, right, as it flows through the ejaculatory duct. What's this? That's the urethra, right, from the urinary bladder. That's allowing for passage of urine. See it? The two merge to form a very short, another short stretch there, which goes through the prostate, and therefore we call this the prostatic urethra. Another important landmark that you want to pay attention to is this. This is called the urogenital diaphragm. So the stretch of tube, the urethra that goes right through just that diaphragm region is called the intermediate part of the urethra, which then, as you can see, this is the last accessory gland. This is the bulbourethral gland, allows for the passage of sperm in all of these secretions from the different accessory glands. It then allows it to pass through the center of the penile tissue, which is obviously the spongy urethra. So when it comes to the male duct system, there's several different stretches. It's a one continuous tube, but it has different names in each different section. And this picture does it some justice, but the models that you'll have in lab, you'll be able to see these different stretches a lot better. Work with the models. It's not so relevant for my lecture exam, but it is probably going to be on that lab exam. And this is one that's always very confusing for students. So just remember, first you have the ejaculatory duct, which kind of merges with the urethra, becomes a short stretch there called the prostatic urethra, and then this tiny little region there, as it goes through the urogenital diaphragm, is the intermediate part of the urethra, and the rest of this tube is the spongy urethra. Okay. Spongy urethra is surrounded by erectile tissue, right? So this darker blue region that immediately surrounds the spongy urethra is called the corpora, or the corpus spongiosum. And then you have a pair of erectile bodies on the outside of that. This is called the corpora cavernosa, which then allows for, so as you can see, the spongy urethra is, what do you call it? It's a shared conduit, right? For both the passage of semen as well as urine, which then gets eliminated from that external urethral orifice right there. Okay. Okay. So then let's talk a little bit more. Okay. Just a little bit more about the structure of the testes. Well, first of all, the testes is housed within that sac-like structure called the scrotum, right? And this is housed outside of the male body because the sperm that are being produced within the testes, they have to be maintained at a few degrees lower than the core body temperature. That is the best for production as well as viability of that sperm, right? So therefore, we've got to find a way to cool blood flow as it reaches those sperm, because that's the best ideal environment for its survival. Okay, there are two muscles, one smooth and the other is skeletal muscle. So the darkest muscle is smooth muscle. Cremaster is skeletal muscle, and these bands of muscle will help to basically elevate or lower the scrotum. So if it is if the core body temperature is a little too high, like on a hotter day, you want to lower the scrotum so that you're not damaging the sperm within the testes, within those seminiferous tubules. You want to lower it. On a colder day, you want to raise it. Okay. When you're looking at okay, let's talk about how you maintain the temperature of those sperm within the testes. So again, looking at this, you've got the testes here, and of course you have the paired structure on the other side. You're seeing a little bit of the epididymis here, maybe the head region. The rest of it is behind the testes. Then you've got the cremaster muscle here, which is kind of helping with the raising and the lowering of the testes, depending on the core body temperature. This entire structure here is called the spermatic cord. So what is the what's inside there, right? Within the spermatic cord, there are four different structures we want to keep in mind. One, let me back up. You'll remember how the epididymis became the vas deferens, right? So the vas deferens is it's inside of that spermatic cord. So let me go back here for a minute. So here's that brown tube. That's the vas deferens. That's for the passage of sperm. As sperm are produced here within the seminiferous tubules, they get stored in the epididymis. And when they need to be released, it's going to pass through the vas deferens, right? What else do you see? Well, you see yellow. Yellow is going to be nerves, because we've got to provide signals, right, to innervate those structures. Then blood vessels. So you got the red. Red is going to be the testicular artery, which is going to carry what? Oxygen-rich blood, nutrient-rich blood to the seminiferous tubules, the testes, and all those organs, because I want to supply nutrients and oxygen to those areas. What about the pampaniform venous plexus, which is this network of blue vessels? That's part of the not just one vein, right? It forms like a network of vessels. You see how it kind of wraps around this entire region here, right? Okay, so think about this. You have testicular artery that's carrying what? Oxygen-rich blood, right? And that is from the core of the body. So that's at that core temperature. And it's carrying that blood towards the testes. So if that blood reaches the testes where sperm are being produced, right? that would be too high. Do you see that? I have to find a way to cool that blood down just a little bit, 2 or 3 degrees lower. This is why the blood leaving out of the testes, which is through the venous plexus, that's at a lower temperature, and it kind of wraps around the testicular artery, kind of drawing all of that extra heat out of it. Does that make sense? So by the time the oxygen-rich blood actually gets to the testes, it's at that right temperature, slightly lower. Okay, that's the purpose of that pampaniform venous plexus. Okay, we're gonna stop there. We will go into details of what's going on within the testes and how do you produce sperm through this process called spermatogenesis next class on Tuesday. So I'm going to tell you this. One of the concepts that I'm going to cover very, very quickly is meiosis. This is very similar to what you covered with mitosis, prophase, metaphase, anaphase, telophase, and all that fun stuff, except there's 2 rounds of meiosis, and this is how we're producing gametes, like the sperm cells, or with the female, similar processes occurring with oogenesis. I am not going to be able to go into the details of prophase, metaphase. All of that was covered. Nothing's different there. What is unique about it is some concepts of recombination. I will talk about that. So I need you to please review, and I'll go ahead and post some videos as well. There's plenty of information out there on meiosis. You want to be prepared, because I'm not going to all those details. Okay, I'm going to talk about what's really unique about it. That's what I need to focus on so I can move on to spermatogenesis. Okay. See you all around. So today's Thursday. Are you still working on male anatomy, or are you starting to work on female? Female, maybe? Oh, good, because I'll need to cover that on Tuesday. All right. Got it. You need something? Oh, picture. Okay, got it. Remote users, I'm going to say bye to you all. Have a wonderful rest of your day.